nucleotides include nitrogenous bases such as adenine, thymine, guanine, and cytosine. A deoxyribose, a phosphate group, would make adenine, thymine, guanine, and cytosine. The DNA double helix was discovered in 1953 by James Watson and Francis Crick. It was discovered by beaming X-rays through DNA crystals and then recording the scattered X-rays with photographic film. It created a cross shape, which is common for a helix-shaped molecule. A hydrogen bond is a dipole-dipole attraction between two nitrogenous bases, cytosine and guanine, and adenine and thymine, which allows the nucleotides to bond, forming the rungs of a twisted ladder known as the DNA double helix. The nucleotides are connected by the phosphate backbone, which binds the number 3 carbon of one deoxyribose to the number 5 carbon on an adjacent deoxyribose. At the end of each nucleic acid chain is either an unbound 3 carbon, the 3' end, or an unbound 5 carbon, the 5' end. The two chains of DNA run anti-parallel to each other. This means that at the ends of a DNA molecule, one chain has a 3' end and the other chain is 5'. DNA replication. Step 1. DNA double helix is unzipped by helicase, an enzyme that breaks the hydrogen bonds holding complementary bases of DNA together. Separation of DNA forms replication fork. A small piece of RNA called an RNA primer acts as a starting point for DNA synthesis. One attaches itself to the end of the leading strand, while several attach themselves at random points on the lagging strand. Step 3. A small piece of RNA called a primer binds itself to the end of the leading strand. It acts as the starting point for DNA synthesis. DNA polymerase binds to the leading strand and moves across it, adding complementary bases. RNA primers are made and bind to random points on the lagging strand. Chunks of DNA known as Okazaki fragments are added to the lagging strand. The Okazaki fragments will join up later. A closer look at the process reveals that there are two enzymes responsible for DNA synthesis, DNA polymerase 1 and DNA polymerase 3. DNA polymerase inserts itself at the replication fork. There, it adds nucleotides on the 5' to 3' leading strand in one long continuous chain and does not stop until it gets to the end. On the 5' to 3' lagging strand, it synthesizes DNA with gaps in between, forming the Okazaki fragments. This process is very fast, with DNA polymerase 3 adding nucleotides at a rate of 1,000 per second. DNA polymerase 1, which is significantly slower, has two functions. It proofreads any DNA made by polymerase 3, correcting any nucleotide mismatches, and it also fills in the gaps between Okazaki fragments. Step 4. After all of the bases are matched up, an enzyme called exonuclease strips primers off of strands. The gaps where the primers were are filled with more complementary nucleotides. Step 5. DNA is proofread to avoid mistakes in the sequence. Step 6. An enzyme called DNA ligase seals up the strands into two continuous double strands. The result is two new DNA molecules with one new and one old strand of
So one of the differences between RNA and DNA is that DNA is two strands while RNA only has one. DNA is much longer than RNA. RNA is actually pretty short. Another difference is that DNA has deoxyribose sugar while RNA has ribose sugar. Another difference is in the nucleobases. In RNA, there is uracil. Meanwhile, in DNA, there is thymine. Yet another difference is that DNA makes RNA, makes proteins. Transcription of DNA to RNA. Differences between the two. Here's a model of DNA and RNA. So one of the differences between RNA and DNA is that DNA is two strands while RNA only has one. DNA is much longer than RNA. RNA is actually pretty short. Another difference is that DNA has deoxyribose sugar while RNA has ribose sugar. Another difference is in the nucleobases. In RNA, there is uracil. Meanwhile, in DNA, there is thymine. Yet another difference is that DNA makes RNA, makes proteins. Different types of RNA. There is tRNA, which is transfer RNA and carries amino acids to ribosomes. There's mRNA, which is messenger RNA and tells the ribosomes what to make. There's rRNA, which is ribosomal and helps the mRNA bind and can also act as an enzyme to speed up reactions. So basically all of this happens in the nucleus. Introns and exons, yay! Basically here in the RNA, the introns get cut out because they don't work with this, and then the exons, which are the remaining parts, get glued together. Making of mRNA message and the leaving of the nucleus. For replication of DNA, it must first get unwound, which happens by an enzyme coming along and straightening it out. After that, we unzip it with the helicase, which is that enzyme. The promoter tells this where to happen, and the RNA polymerase is the thing that makes the bases match up. At the end of this, there's a terminator, which tells the enzymes to stop working. Then, to finish this off, you just add a poly-A tail and a 5 cap.
Genetic code and how to decipher it. Genetic code allows DNA and RNA to be decoded into proteins. It's decoded in groups of 3 called codons and specify an amino acid. Protein creation starts with a start codon and ends with a stop codon. After the 3rd stop codon, the protein is made. The reading direction depends on the ends of the DNA or RNA, which are the 5' end and the 3' end. Protein creation starts with initiation, which start with the mRNA ribosome and an initiator tRNA gathered by the initiation factors. The small and large ribosome subunit envelop the mRNA to start translation, which matches corresponding amino acids to create a polypeptide. The tRNA carrying methyl 9 attaches to the small subunit, binding to the 5' end of the mRNA. They move towards the 3' end until it reaches the start codon. We are now on to elongation. Inside the ribosome are 3 areas, site A, P, and E. So ape, very much ape. The first methionine carrying tRNA starts in site P. Site A will have an anticodon that's complementary to the first. rRNAs inside the ribosome creates a reaction and the 2 amino acids connect, turning into a peptide. It also transfers the methionine from the first tRNA to the amino acid of the second. The mRNA is pulled forward by 1 codon, allowing the now empty first tRNA to exit through site E. A new codon is exposed in site A and the process repeats. That is, until termination, where the finished polypeptide is released. That happens when a stop codon enters site A. It is recognized by proteins called release factors. It adds a water molecule to the last amino acid of the chain, thus separating. Afterwards, the small and large ribosomal subunit also separates from the mRNA and each other to take part in another round of translation. Meanwhile, the newly released polypeptide folds together and creates a protein with a mustache.
3 types of mutations are frame shifts, substitutions, and inversions. A frame shift mutation is a mutation that changes how the codon sequence is read, such as an addition or deletion. A substitution is a mutation where one nucleotide is replaced with another nucleotide. An inversion is a mutation where one set of nucleotides is inverted to create a new order. Different mutations can cause different problems with the structure and function of a polypeptide. Silent mutations are mutations that have no effect on the structure or function of a polypeptide.
Mutations and albinism. Albinism is a frame shift mutation. This is the normal process. The polypeptide created is pretty long. This is the mutated process. The polypeptide created by the mutation is much shorter. When made into a protein, the shape is weird and it can't function properly, thus resulting in pale skin, eyes, hair, and susceptibility to sun. Yeah, you learned a thing.